Hello everyone, welcome back to another video from Shomu's Biology. In this series of lectures, we are talking about different parts of immunity and immune system, and today we are going to talk about the 5 major antibodies of our immune system. What are these antibodies? What are their structures? We are going to talk about the functions of these antibodies, and we are also going to talk about the clinical relevance of antibodies in our body. So let's start with the first one, the overview of antibodies. Starting with the antibodies or immunoglobulins, they are glycoproteins produced by plasma cells. So plasma cells are antibody secreting factories. B cells are modified into plasma cells upon activation, and these plasma cells start producing glycoproteins, which are known as immunoglobulins or antibodies. So these glycoproteins means they are primarily protein, but they have carbohydrate moieties present in their structure. Key role in immune defense: neutralizing the pathogens, activation of immune cells, and also opsonization of the pathogens via phagocytosis. When the antibody coat a pathogen, then the phagocytic cells like macrophage, neutrophils, dendritic cells can engulf that pathogen more easily. That is known as opsonization. That is their purpose. 5 major types are IgG, IgA, IgM, IgE and IgD antibodies, and you can see all of them are listed in here, where IgG, IgE and IgD are all Y-shaped single single antibodies, and they have 2 Fab region where they interact to the antigen, and this is Fc region where they interact to the receptor on the surface of different immune cells like T helper cells. So Ig here, IgG, IgE and IgD, these are Fab regions. Okay, remember that. So there are 2 Fab regions. So for each antibody the valence is 2. For IgG, IgE and IgD the valence is 2. But IgM can be found as a pentamer, where 5 into 2 you get a valency of 10 for IgM, where for IgA sometimes found as dimer. So we have 2 into 2 valency of 4. So 1, 2, 3, 4. This is very important. And as the IgM has highest valency to bind to the antigen, because IgM as a pentamer can bind to maximum antigen, so antigen antibody complex formation is mostly done by IgM antibody. An antigen antibody complex formation is required for fixation of the complement. So for complement fixation, IgM is predominant, while IgA is present in the secretions of our body. So they are also known as secretory antibodies. Rest are something that we are going to see one by one now. First is IgG. IgG is the most abundant antibody in our body. Okay, structure is a monomer with flexible hinge region in it. You can clearly see, like any kind of antibody. I am not going to explain this for every single antibody, but once for IgG, that the antibody, you can see 2 separate chains. One is in the purple color, Y shape, this one, 2 chains connected here by disulfide bond. Okay, so these are protein chains, and there is also green color chains. The green color chain is known as light chain because it is smaller in size, so the molecular weight is less, and this purple color chain is bigger, so they are known as heavy chain. They are heavy chain. And you can see in the top, in the Y-shaped structure, in the top part, this is Fab region. In the bottom we have Fc region. Okay, and what else we know is that till this section, so a part of heavy chain, a part of light chain is known as constant region, means that part of the antibody remains the same, there is no change in it, and 1/4 of this is 1/4 of heavy chain, and half of the light chain that we see here and here is known as variable region, means the sequence, antibody sequence, the amino acid sequence there in those portions of this antibody will vary, and they continue to vary, and variation in those region leads to the change in the IgG. Even all IgGs are not same, because the variable region of the IgG are different. okay, and based on that there are different subclasses. And why it is important? Because this FAB region is the place of antigen binding. This is carrying antigen binding site. One site binds to one antigen at a time. So for two sites it will bind to two antigen. So for a single monomer like this IgG, which is a monomeric antibody, two antigens can bind together. So the valency will be provided as 2. Remember, the valency is 2 for IgG, IgM, IgD. IgG, IgE and IgD. For IgM, the valency is 5 into 2, 10 pentameric, and for IgA it is 4, 2 into 2, because it's dimeric. Okay. So now let's move to the subclass. IgG have multiple subclass: IgG1, IgG2, IgG3 and IgG4. Functions: long-term immunity. The IgG provides a long-term immunity. It also have function of opsonization and toxin neutralization. So neutralization and opsonization is functions provided by most of the antibodies, but predominantly IgG provides long-term immunity. IgG is an antibody of our secondary immune response, and in secondary response they produce more and more IgG to fight against pathogen or specific antigen. And as there are variable regions, they can alter the sequence of amino acid in the variable region, so that from one particular DNA, one particular gene, they can produce multiple varieties of IgGs, multiple types of IgG proteins, and those multi types of IgG proteins have the capability to bind with the antigen with specificity, because there are numerous combinations based on the type of antigen that we encounter. Our body is producing so many varieties of antibody, hoping that one antibody from those huge pile of antibodies will bind to the antigen. Our whole immune response is based on this same idea, that if we are encountered with an antigen, at least one part or one category of antibody is going to bind to it as a lock and key fit. And if at least one antibody binds, that antibody production will be triggered and the cells switch to production of that kind of antibody very soon, within hours to days, and then they start fighting against the infection, then they start fighting against the antigen and the antigen releasing pathogen. That's what we depend on, that's what our immunity depends on. Okay, numerous combinations are there, that's why we have the variable regions in antibodies. In all antibodies we have the same idea. We have two chains, two heavy chains and two light chains. We have the variable regions as well as the constant region. Constant region is known as FC region, C for constant, this one. Okay, and variable region is in the top, where 1/4 of the heavy chain and half of the light chain is present in the variable region. Next, the function is activating complement pathways and cross placenta for passive immunity. Another very important feature of IgG antibody is that it can cross placenta and transfer from the mother to the fetus to provide immunity to the growing embryo. Clinical relevance: elevated levels of IgG signifies infection, active infection in the body. So we always measure the antibody titer, we measure IgG titer. If the IgG values are high, that means active infection is going on in our body. Used in the passive immunotherapy as well. Second type of antibody is IgA, Immunoglobulin A. It provides mucosal defense. So if you look at the structure, it is also monomeric in serum, but when it is secreted, it becomes dimeric. In the secretions, in the saliva, in the tear, in those secretions, it is dimeric, but in the serum, in the blood, it is monomeric. Okay, so what we have here, we have this two, this is 1 and this is 2. Okay, and both of them are connected by a protein chain known as the J chain, and the J chain is connected to FC regions of the IgA or immunoglobulin A. Okay, and they also have the light chain here. This is light chain, you can see in the light blue color, and the heavy chain is the dark green color. You can see that they are connected by the J chain here. Okay, and J chain connection is really important, because J chain connection helps the IgA to be connected during secretion. In the secretory format, we need the dimeric form, not the monomeric form. Okay. Subclasses. Immunoglobulin A has two subclasses, IgA1 that is present in the serum and IgA2 that is in the mucosal secretions. Functions of IgA: protects mucosal surfaces, that is respiratory tract, gastrointestinal tract are being protected by this IgA. They are patrolling there. So if there is any antigen, they are going to neutralize it or can initiate opsonization in those tissues. Prevents microbial adherence to the surface of our cell. Provides neonatal immunity via breast milk. Yes, not only in the saliva or tear, but the IgA is also found as a dimeric form in the breast milk. So obviously it protects the neonates because with the milk they get their required immunity boosters. Clinical relevance: deficiency of IgA linked to recurrent infections in the body, elevated levels found in the mucosal infections. If any gastrointestinal mucosal infection due to infection or due to diseases like IBS, IBD, there can be elevated levels of IgA seen there. So now IgM, the first responder, IgM is known as the first responder and this is pentameric in serum. So when IgM is present in our blood in the serum, it is pentameric in nature. So you can see 1, 2, 3, 4, 5 IgM, they are connected together by the separate bonds out there in the J chain that is out there. Okay. So pentameric means one IgM bind to one IgM antibody binds to 2 antigen. So 2 into 5 such monomers, total 10 antigen can be adhered or attached to the antibody. Okay, that is very important. Subclass is not identified. Functions: first, antibody production in the immune response. The first set of antibody that is being produced in the immune response is the IgM. So IgM is the antibody of primary immune response. This is the antibody of primary immune response. Okay, by default our B cells have B cell receptor on surface, that is, those are IgD antibodies, but then when they are getting activated they start producing IgM antibody. IgM antibody, why? Because they are the primary immune response antibodies. They start being producing IgM antibodies and they are the first responders. They activate complement pathway in many folds because for the activation of complement pathway the antigen antibody immune complex formation is very important, and immune complex means antigen antibody complex, and when there is antigen antibody complex there is a chance that the complement proteins can activate and can fix complement, can cause pores to the target cell and can damage the target cell, target tissue. That can be done. Okay, so this is very, very important. Remember, the antigen antibody complex formation can be triggered by this IgM. Okay, because it is pentamer, it helps to do that quite easily. It activates the complement pathway and effective against pathogen agglutination, because as it can cause the immune complex to be fixed by complement activation, it can cause agglutination. Pathogen agglutination can be done, which can prevent the spread of the pathogen. Okay, clinical relevance is that the IgM concentration is elevated in any kind of acute infections, acute, not in chronic infection. In chronic infection, you will see, in chronic infection, you will see more IgG being produced because IgG antibody is an antibody of secondary immune response, while IgM is an antibody of primary immune response. So they are found in the acute infections, and diagnostic markers used for recent infections. For recent or current infections, we use IgM as a marker. If it is there in your bloodstream in high concentration, we can comment that yes, there is acute infection going on. Let's move to the fourth category, IgE category. IgE antibody is known as mediator of allergies because IgE causes the type 1 hypersensitivity. Not cause, but actually IgE's response is there in terms of type 1 hypersensitivity. It's a monomeric form with an extra constant region for mast cell binding. That's why we can easily distinguish between IgG from IgE, because IgG and Ig is very much similar. The only difference is that IgE has one extra part to the constant region, one extra fragment to the constant region. You can see that generally the constant region is ended with two sections, but in IgE they have an extra part. So in the constant region we have CH1, CH2, CH3 and CH4. CH4 is the extra part. Okay. So this is variable region, VH for variable heavy chain, VL for variable light chain. Okay, that's it. How many domains? 5 domains. Molecular weight is mentioned. How much carbohydrate is present in Ig? 12%. The half-life of Ig in the serum is 2.5 days. These are some information that you may remember, may not remember, that's your choice. But remember one thing, that the Ig has one extra constant region to bind to the mast cells, and this is the part that helps this Ig to bind to the Fc receptor that is present in the surface of mast cells. Okay. Subclasses, no subclasses identified. Functions: trigger mast cell and basophil degranulation, causes the granules that are present in the mast cell to be fused in the membrane and release of granular component into the tissue. Mediate allergic reactions such as asthma, anaphylaxis. The type 1 hypersensitivity like asthma, like anaphylaxis is caused by IgE. Defense against parasitic infections. Defense against helminths are also being provided by IgE by activating antibody mediated cellular cytotoxicity. So if there are bigger helminth structures, this IgE will cover the helminth surface, and then they will recruit further immune complex like complements and other complexes, or the phagocytic cells can also destroy, but the complement activation causing pore in the membrane of the helminths and can destroy that. Clinical relevance: IgE is elevated in allergies and parasitic infections. So to assess allergy, type 1 hypersensitivity, generally the blood test is done to check for the levels of IgE. Next is the IgE targeted in allergy therapies, like anti-Ig antibodies are used if somebody is allergic due to hyper IgE production in the body. If your body has more IgE production, in that case we can use anti-Ig antibodies to block the effect of IgE or Ig antibody. Last one is IgD. IgD is the fifth type, okay, and the least important among all this is known as B cell activator, IgD. Okay, it is monomeric and it is also very similar to IgG. You can see the IgD structure. It has CH1, CH2 and CH3, 3 separate components of the constant heavy region, and they have a variable heavy region, they have a variable light region. The light chain is denoted with the light blue color and the heavy chain is denoted with the green color. Okay, heavy chain is known as delta, light chain is known as kappa or lambda. Okay, so now the subclass not identified, acts as an antigen receptor or on immature and mature B cells. So again, B cell receptor, IgD also act as this B cell receptor. Remember I told you IgM and IgD. IgD also act as a B cell receptor. The default B cell receptor plays a role in B cell activation and regulation. So if you tell me what is the role of B cell receptor by IgM, by IgM or by IgD, I will say IgD, not IG. IgM can also act as a B cell receptor, but IgM is a default antibody that is produced, the default antibody that is being produced by the B cell, that is IgM. But IgD is the antibody that is always being present and present in the surface of the B cell. So remember, if you recall our lectures of immunity, you will remember that we have talked about B cell receptors quite a lot. So this B cell receptor is nothing but IgD that is present in the surface of the B cell, that interacts with the antigen and can activate the B cell. The clinical relevance is that dysregulation of the IgD can cause autoimmune diseases and associated with chronic lymphocytic leukemia. Okay, these are the clinical relevance of IgD. So IgD is nothing but B cell receptor, the default antibody that is present in the surface of B cell, that is IgD for you. That's all, that's it. Okay, the structure is very similar to IgG, but IgG is produced after the secondary exposure of the body to the antigen, because IgG is the antibody of secondary immune response, but IgM is an antibody of primary immune response. Comparison of the different antibodies for you now. The IgG provides long-term immunity, antibody of secondary immune response, and it is most abundant antibody anytime in our body. Next is IgA. Mucosal, they are present in the mucosal surface and secretions like tears, saliva, breast milk. IgM, initial immune response is provided by IgM and they can activate complement. Okay, complement activation is done by IgM only. IgE cause allergies and is also required in parasitic defense. IgD is nothing but B cell receptor. It helps in the activation of B cells. Okay, so that concludes our understanding of different antibodies, namely IgG, IgA, IgM, IgE and IgD. Okay, and among these five, IgM is pentameric in the serum and IgA is dimeric in the secretions. I believe you like this video. If you like this video, please hit the like button, share this video with your friends and subscribe to get more videos like this in future.